Hello, it's been a while since we've had a video, so why not have a new one? This particular unit is going to be covering cell transport, and basically what that means is how do substances go from in and out of the cell? And we know at this current moment that the plasma membrane is responsible for determining what comes in and what comes out of the cell. But there's a lot more that goes into it than just saying, yes, you can come in or no, you cannot come in. So there's a lot more, players in the game when it comes to the regulation of substances passing in and out of the cell. We're going to talk about cell transport and how it relates to homeostasis. And if you don't remember, homeostasis is the idea that the internal environment of an organism is stable at all times. And our cells play a huge role in this because if they don't have the nutrients and the substances they need, they can't fully function to their jobs, and then this would take us out of homeostasis. Before we start talking about cell transport and what it looks like for cells to have substances travel in and out, I want to take a minute to discuss some basic vocabulary and what a cell would truly look like in various scenarios. So the first word that I want you to be able to define is the word solution. And basically a solution is a physical mixture made of multiple things. And I'm not going to get into a lot of detail. If you go on to chemistry, we'll get into more detail with solutions, but you need to know it's a physical mixture. So our bodies are a large mixture of a bunch of things, and that includes our cells. And on a smaller level, our cells individually have different solutions in them as well that help maintain the functions of those cells. So in order for these cells to maintain their functions and to be healthy, they must have substances coming in and out. Now, when there are issues as in like this, the body is dehydrated, your cells become dehydrated. So the representation of the bigger organism, as in I, as a human, am dehydrated today, means your cells on the individual level are also experiencing a lack of water. And your cells actually look different when they're dehydrated versus when, let's say, you eat a lot of sugar one day. So a couple of terms that I want you to be able to know and be able to distinguish are the words isotonic, hypotonic, and hypertonic. And I'm going to talk about all three of these in this case, but I will spend a little more time talking about them in class. Here on the slide, you'll see that I just included a picture. And if you notice anything, the hypertonic cells look very much skinny and shriveled up versus your hypotonic cells that are much larger. They don't look as donut shaped. They look very round and they could burst. And then your isotonic is that nice happy medium. When we use these words, we're actually talking about the concentration inside versus outside of a cell. So the first type of cell I'm going to talk about is an isotonic solution. So isotonic, if I were to say something is isotonic, that means the solutes or the substances within the solution is the same on both sides. So if I'm talking about a cell, the inside of a cell, there's the amount of solute, or let's just say stuff, the amount of stuff inside is equal to the amount of stuff outside the cell. So that way we can easily transfer. This is nice equal space. This is where you want your cells to be. That means that the body is nice and homeostatic. Everything is maintained. Things are moving and going out as they should be. So equally moving in and out. Hypotonic, on the other hand, is when the amount on the outside of the cell is lower than inside. I want you to think of hippo, very bloated, very large cell. So these cells look very bloated because there's a lot more stuff inside than there is outside. I want you to think of Kool-Aid when we talk about this, and I'll go into more detail here in class, but when we make Kool-Aid, we use a specific amount of sugar. You can use more sugar, but you're going to find your Kool-Aid's going to be a lot more sugary, and then some of it may not even dissolve into the water. Whereas if you don't use enough Kool-Aid, or if you don't use enough sugar, or you don't use any sugar at all, the solution is going to taste much different. So you can create a hypo or hypertonic solution based on how much stuff you're putting in. And that's called concentration. So a higher amount of stuff means something is higher concentrated. Conversely to hypotonic is hypertonic. Hypertonic is when the outside of the cell has a higher amount of solutes than the inside. So again, hypotonic, there's more inside. than outside. Hypertonic, there is more outside than there is inside. So these cells actually look very shriveled, not very happy, because your outside environment much more larger, so we're trying to even things out. And basically what this means is your cell's just going to be constantly passing back water back and forth to even out the amount of solutes, or the amount of stuff. So I keep saying stuff. The true word for that is solutes. We are going to spend several minutes or a lot of time on the idea of solutes and solvents, solutions, and concentration, because all of these words might seem foreign to you, and that's okay. We're going to talk about them much more detail in class. Now let's get back to talking about the transport across the membrane. So remember in the last slide I talked about a solute and how a cell looks if things are transferred in and out, but how does the transferring actually happen? We know that the cell membrane regulates what comes in and out of the cell, but what is actually doing that movement? Now, there are two different ways that things can move across the membrane. They can either go passively or they can go actively. When passive transport happens, and that's what we're going to spend the rest of this video talking about, passive transport. Passive transport involves no energy. So things are moving back and forth using no energy for the cell. This is good because we don't want to use energy for just anything. If it's a little job that can be done without energy, that's a good thing, or we'd be really tired. All right, so passive transport occurs when substances cross the plasma membrane without any input of energy from the cell. We are going from areas of high concentration to areas of low concentration, meaning there's a lot of sugar on one side. I need to even this out, so I'm going to move some of that sugar that's in the high spot over to the low spot. You can think of passive transport like a ball on top of a hill. It doesn't require energy because the ball's going to roll down the hill. That's just the nature of gravity, just like in this instance. So if I have a hill here and I want to get to the bottom and my ball is sitting right at the edge, it's easily going to fall down on its own accord, not using energy. That is passive transport, where we can move things across. We're not using energy. They just freely flow. We don't really see this in many substances. Typically, passive transport happens with low or small substances. And remember, the biggest thing here is we're going from high concentration to low concentration. So meaning we're going from a lot of particles on one side to like very little particles on the other. Always moving from low to high. There are three types of passive transport that you're going to learn about in this video: simple diffusion, osmosis, and facilitated diffusion. All three are similar in the fact that they all involve high to low concentration. They do not require energy, but there's some different factors that come into play and who actually qualifies to transfer across the membrane with simple diffusion versus osmosis. Okay, so simple diffusion, or just shortened up to diffusion, is the movement of a substance across a membrane due to a difference in concentration without any help. So it's a lot of words. Basically, I'm just saying the same thing I already said for passive transport. We are moving substances across the membrane. We're going from high to low concentration. We're not using any help. Things just move back and forth. This simple movement occurs due to the high concentration, like I already told you, from one side to the other. Typically, you're not going to find many things that are going to pass through the cell with simple diffusion. You're going to find things like very small hydrophobic molecules. So pretty much just really small things like carbon and hydrogen. Very, very small molecules. Conversely, we have what's known as osmosis. Now, osmosis is exactly the same as simple diffusion, for one exception. It only involves water. So we are only diffusing water across the membrane. So osmosis is only special because we're just transferring water. So it's the exact same as simple diffusion. It goes from high to low. It moves things across the membrane, but it's just moving water. It's not moving anything else. And we want water to be equal on both sides because this is what creates an isotonic solution, an isotonic cell, and that's what we want our cells to look like. Nice, happy cells that have a good shape. The third type of diffusion is known as facilitated diffusion. And if you think of the word facilitated or a facilitator is somebody that helps move things and makes things go. So if I'm a facilitator of my classroom, that means I'm moving the lessons along. I am guiding the classroom. just like in facilitated diffusion. So we're still dealing with diffusion that does not require energy. We're still going from high to low concentration, but we just need a little more help to get across the membrane. So in order to get some help, we're actually going to use some friends. Those friends are transport proteins, and transport proteins are used to move substances across. So it's like if I want to cross a bridge, but I am too scared to cross that bridge by myself, Barry's going to hold my hand and we're going to cross the bridge together. He is my transport protein. He ensures I get across that bridge safely. We're going to learn about two types of transport proteins in this class. One of them is known as a channel protein. It's like a pore. So if I have the membrane here, you can think of a channel protein like just a small opening into the cell. Very minimal. It's not super big. It's super tiny. It allows large hydrophilic molecules to pass right on through. They don't have to worry about messing with the tails of the plasma membrane or the lipid bilayer, so it's very tall, tiny little holes in the membrane that allow these things to come through. That's one type of transport protein. The other type of transport protein is known as a carrier protein. So something that physically moves something along with it. So what's going to happen, this carrier protein is just going to hang out. They just hang out on the plasma membrane. If I had a plasma membrane here, we would have just, let's say this is a carrier protein. This carrier protein is just hanging out on the cell membrane, and then what's going to happen is we have these tiny molecules that are going to come in and they're going to see this carrier protein. This carrier protein is going to detect, yep, you are who I need. So it's going to bind to each other. So the molecules are going to bind to the carrier protein. Once the carrier protein knows, yes, this is who I need to bring across inside the cell, it's going to change its shape. So it's going to go from something like that to something like this, along with the molecules in there. Now, because it changes its shape, it can move these molecules across into the cell. So this is the outside. So we're going to move these molecules into the cell. After that, the carrier protein goes back and it just goes back to its regular shape as before, but it has to detect. It's a really beautiful relationship because it has to be a very specific molecule to attach to this carrier protein in order to bring things in. So really nice. So that is our second type of transport protein. Both of these proteins, again, are responsible for helping move things along from high to low concentration. So that is pretty much it for passive transport. This is a lot of information. I probably talked really fast. So if you need to rewatch, please do so. If you have any questions, please make sure to ask me on Edpuzzle. Thanks.